Good afternoon, students. So, today we are going to study the remaining part of lipid metabolism. So, these are the topics we are discussing today: de novo synthesis of fatty acids, degradation of cholesterol, then some disorders. In that, I am discussing only one disorder. Remaining disorders we can discuss in next classes. So, de novo synthesis of fatty acids. See, de novo synthesis of fatty acid means this is happening in liver, kidney, adipose tissue, mammillary glands, etc. So this production takes place in cytosomal fraction of the cell. So it takes place in liver, kidney and adipose tissue, etc. And the process or the production takes place in the cytosomal fraction of the cell. That is, here the two things are important. One is acetyl CoA, another one is NADPH. These are very important. They plays very important role in this synthesis. This acetyl CoA means they are the source of carbon atoms, and while NADPH means they are the reducing agents, and ATP is also very important, and this supplies us energy for these activities. So here you can see three stages. One is production of acetyl CoA and NADPH. Second step is conversion of acetyl CoA to malonyl CoA. And third one is the reactions of fatty acid synthase complex. So this is the first step. That is the production of the acetyl CoA and NADPH. Here you can see from mitochondrial matrix, the pyruvate. These are the starting, that is pyruvate, and they are going to reacting with pyruvate carboxylase, and here pyruvate dehydrogenase. So while reacting with this pyruvate carboxylase, we are getting oxaloacetate, and when reacting with pyruvate dehydrogenase, we are getting acetyl CoA. So this acetyl CoA are very important for the fatty acid synthesis. Actually, this is producing in mitochondria. You can see this is mitochondrial matrix. So here acetyl CoA is producing. How acetyl CoA is producing? That is by the oxidation of pyruvate. That is the pyruvate dehydrogenase. Also the fatty acids oxidation and amino acids. There is degradation of amino acids. This is how the acetyl CoA is formed. And from ketone bodies also we can form acetyl CoA. Sometimes mitochondria are not permeable to acetyl CoA. So here we are using an alternative pathway or alternative arrangement. So this acetyl CoA will transfer to cytosol because they are impermeable. So we cannot stay in mitochondria. So they are moved to cytosol. For that, we have to undergo certain steps. So this acetyl CoA and this oxaloacetate are reacting with citrate synthase, and we will get citrate. So this is how they are moving to cytosol. So you can see citrate here in cytosol side. So this citrate again react with citrate lyse. They form oxaloacetate. In between, ATP produces energy. That is, ATP gives us ADP plus PI, and the coenzyme produces acetyl CoA. And this oxaloacetate react with malate dehydrogenase, and we are getting malate. So in between one reaction takes place, that is NADH plus H positive versus NAD positive. Then this malate will react with malic enzyme and finally they'll form pyruvate. In between certain reactions are taking place in here. That is NADPH is generated. Also carbon dioxide is generated. You can see here. And they result in fatty acids. So this is the production of acetyl-CoA and NADPH. This is the explanation of this. Then conversion of acetyl-CoA to malonyl-CoA. This is the second step. So this is very important step. So here you can see acetyl-CoA is carboxylated to malonyl-CoA. So for that here we are using acetyl-CoA carboxylase. So this is an ATP dependent reaction. For that we require biotin. See, biotin is mainly for carbohydrate fixation. So this is the conversion of the acetyl-CoA to malonyl-CoA, or the formation of malonyl-CoA. Using acetyl-CoA carboxylase we will get malonyl-CoA. And the third step, that is reaction of fatty acid synthase. So these reactions takes places by FAS complexes. So for example, you can say in eukaryotic cells, the fatty acid synthesis will exist as dimer, and they having two identical units also. So each monomer, they possess different types of enzymes and one ACP, that is acyl carrier protein, and this ACP will bound to 4-phosphopentadiene. So again I'll repeat, so in eukaryotics this synthesis exists as dimer. So these dimer have two identical units. So there is each monomer, they have different types of enzymes. It is enzymes, and there will be ACP, and this ACP will bound to 4-phosphopentadiene. So these fatty acid synthase will catalyze these reactions. That is, they are having 7 different enzymes. So they will catalyze all 7 reactions. So when this complex is dissociated, they results in the enzyme activities. There is loss in the enzyme activities. This is in the case of eukaryotics. That is in the case of prokaryotics, means they are carried out by the multi-enzyme complexes and with a separate ACP. So ACP is very important in the fatty acid synthase. So let's go through the reactions. You can see they'll start with acetyl-CoA. This is FAS complexes. And acetyl-CoA will react with acetyl-CoA ACP transacylase. And with coenzyme, we will get acetyl-S-ACP. So this is the first step. That is, acetyl-CoA is converted into acetyl-S-ACP using acetyl-CoA ACP transacylase, and with the help of coenzyme, also with the help of FAS complexes. And from this acetyl-S-ACP, the acetyl group is transferred to cysteine. This is the second step. Let's see. You can see here acetyl group is transferred, and also we'll get malonyl CoA. And these both will react with malonyl CoA ACP transacylase, also with coenzyme, and we will get acyl malonyl enzyme. And this acyl malonyl enzyme will react with beta ketoacyl ACP synthase. Also here carboxylation takes place. We will get beta ketoacyl ACP. Then this beta ketoacyl ACP will react with beta ketoacyl ACP reductase. So here reduction occurs. This is the reaction. And we will get beta hydroxy acyl ACP. This is the reaction. Then next step. This beta hydroxy ACP, they'll undergo dehydration using beta hydroxy acyl ACP dehydratase. So we will get trans-enol ACP. Then this trans-enol ACP again react with enol ACP reductase. Reduction occurs and they'll form butyryl ACP. Butyryl ACP. Then from this transfer of carbon chain from a butyryl ACP to cysteine. So you can see this is acyl S enzyme. So this reactions is repeated more times. It is 6 more times, and finally we are getting this. This is the repetition, and using thioesterase we are getting palmitate. So this is the de novo synthesis of fatty acids. So the reaction begins with acetyl CoA and ends with palmitate. So this is the overall reaction of the synthesis. You can see 8 acetyl CoA, 7 ATP and 14 NADPH plus 14H, and finally we will get a palmolate coenzyme 7 ADP plus PI 6H2O. So in palmitate, out of these 16 carbon atoms, only 2 carbon atoms come from acetyl CoA and remaining 14 carbon atoms coming from malonyl CoA. Then fatty acid synthase complexes. So this is based on the work of Wackell. And we already discussed that they are in the form of dimer and they are having 2 identical monomers. So the molecular weight is this. That is 2,40,000. Then each monomers, they have 7 enzymes. Also with ACP, and they will bond with phosphopentadiene. So you can see the picture here. FAS complexes. So this is a dimer and they're having 2 identical subunits. See, you can see 2 identical subunits. These are the 2 subunits. all having 7 enzymes. These are the 7 enzymes. And they will react with seven enzymes of FAS, and they will linked with ACP with 4-phosphopentethene. So this is 4-phosphopentethene they are linked with. So you can see here ACP molecule. And these two subunits, this is subunit division, they are parallel, anti-parallel to each other. And this group, that is SH group of this phosphopentethene, is very close to SH group of cysteine. Then each monomer of this FAS, they contains enzyme activities of fatty acid synthesis. This is the each monomer. They have particular activities. So this dimer is functionally active. Why it is functionally active? This is because they consist of half. That is half subunits. I told they have two identical subunits. So they are interacting complementary. That is why they are functionally active. Then this FAH structure, this having both functional division and subunit division. So this is subunit division, and you can see this is functional division. And these two functional subunits, they are independently operate and synthesize the fatty acids. So we can see the functional significance of these complexes. So they are having greater efficiency. And this entire process is confined to a complexes, not out of complexes, they are confined to complexes, and there will be no impermeability barriers for the different intermediates. Then multi-enzyme polypeptide complexes, they are coded by single genes, and there will be good coordination for the synthesis of these complexes. So they having great efficiency, and these whole process are confined in a complexes, and there will be no permeability for each intermediates. And this polypeptide is coded by a single gene, and there will be good coordination for the synthesis of the enzymes in the complexes. Next is cholesterol. See, cholesterol is exclusively found in animals. We can call it as animal sterol. So total body weight in a man is 70 kg, means it's about 140 gram. That is, in our body, some part is cholesterol. It is around 2 kg body weight. Then cholesterol is amphipathic in nature, and they possess hydrophilic and hydrophobic regions. Then biological significance of the cholesterol. See, they are very important component for the cell membrane, and they helps in the synthesis of the many steroids in our body. Example, steroid hormones is vitamin D, bile acids, etc. And these are very important for the lipoproteins, the structure of the lipoproteins, in order to form lipids transportation. Next is fatty acid are transported to liver with the help of this cholesterol esters, that is for the oxidation. So this is the significance of the cholesterol. So let's move to a very important part, that is degradation of the cholesterol. So 50% of cholesterol is converting into bile acids. So they serves as, we already discussed that, for the synthesis of steroid, vitamin D, coprestol, cholesterol. Cholesterol helps a very important role. So you can see this is cholesterol. So from cholesterol, the three compounds, it is pregnenolone, 7-dehydrocholesterol and 7-hydroxycholesterol. So from pregnenolone, progesterone is formed. So from here, androgen, androsterone, testosterone, estrogens, estradiol, estrol, estrone. So these are the steroid hormones. And here 7-dehydrocholesterol, we can see vitamin K. And some corticoids is also formed, mineralocorticoids, adrenaline, corticosterone, glucocorticoids, cortisol. Here from 7-hydroxycholesterol, primary bile acids are formed, secondary bile acids are formed. So primary bile acid means chenodeoxycholic acid, cholic acid. Secondary bile acid means deoxycholic acid and lithocholic acid. And finally ursodeoxycholic acid. So synthesis of bile acids. Bile acid possesses 24 carbon atoms, also hydroxyl groups, that is 2 or 3 hydroxyl groups, that is in the steroid nucleus. And there will be side chain, inside chain there will be a carboxyl group. They are amphipathic in nature, that is we also discussed in cholesterol. Then there will be polar and non-polar groups, and they act as a emulsifying agents in the intestine. Then they also participate in the digestion as well as absorption of the lipids. So the synthesis of the primary bile acids that takes place in liver. And this is the reaction. See, this is cholesterol. Cholesterol with the help of 7-alpha hydroxylase, we will get 7-alpha hydroxycholesterol. So this 7-alpha hydroxycholesterol results in the formation of the bile acids. This is primary bile acids. Cholic acid, chenodeoxycholic acid. Next is secondary bile acids. That is from cholic acid, they will react with glycocholic acid and taurocholic acid. We are getting deoxycholic acid. And here glycokenodeoxycholic acid, taurokenodeoxycholic acid reacting, we will get lithocholic acid. Cholic acid and chenodeoxycholic acids are primary bile acids. And these bile acids on conjugation with glycine and taurine, with the help of conjugation reaction, they will form deoxycholic acid and they will undergo enterohepatic circulation. And lithocholic acid will undergo to phases. Enterohepatic circulation means these bile salts, they will synthesized in liver, that is in gallbladder, and they will secrete it to small intestine. And these agents or emulsifying, they act as an emulsifying agents, that is for the digestion and the absorption of fats and vitamins. And the major portion of bile salts, they are reabsorbed and returned to the liver, that is through portal vein. And these bile salts are recycled and it is reused in several times in a day. This is known as enterohepatic circulation. So enterohepatic circulation means these salts will synthesized in liver and they are accumulated in gallbladder, and this will secrete towards the small intestine, and they also act as emulsifying agents for the digestion and absorption. That major portion will reabsorb and return to liver, that is through a portal vein. And these bile salts, they are recycled and reused many times in a day. Actually, this is called enterohepatic circulation. So about 30 gram of bile acids secreted in an intestine each day, and 0.5 gram, that is lost as a phases. Then choleutesis. So the bile acids, salts or phospholipids, these are the two reasons for keeping cholesterol in a soluble state. So due to the deficiency of bile acids, this condition occurs, it is cholesteatosis. They will precipitate in gallbladder and they result in cholesterol gallstone diseases. This is mainly due to many reasons. Why? One is defective absorption of bile salts from the intestine. Another one is impairment in liver function. Third one is obstruction of biliary tract. So, remedy is administration of bile acids, chenodeoxycholic acid, commonly known as chenodiol. Then synthesis of steroid hormones is from cholesterol. So we already discussed that cholesterol is a precursor for the steroid hormones. So these are the steroid hormones: glucocorticoid, mineralocorticoids, progesterone, androgens, estrogens. So cholesterol will convert into pregnenolone, progesterones, and again they divided into glucocorticoids, mineralocorticoids, androgens. Androgens is turned into estrogens. So these are the synthesis of steroid hormones. Then synthesis of vitamin D. 7-dehydrocholesterol. This is used as an intermediate in this synthesis, and they converted into vitamin D3. So this is cholesterol. So cholesterol is converted into 7-dehydrocholesterol. Then they will result in vitamin D3. So this is the synthesis of vitamin D. Next is hypercholesteremia. What is hypercholesteremia? It is the high level of cholesterol in our blood. That is called a hypercholesteremia. Here HDL, non-HDL and LDL, they will be very high during this condition. It is hypercholesteremia. A normal range of cholesterol is 140 to 200 mg per dl. That is below 200 means it is okay. Border is 200 to 239 and high means it is above 240. What are the causes of the hypercholesteremia? Sometimes it is inherited. Maybe your grandparents will have, so it will transfer to your father like that. Sometimes liver may produces too much cholesterol because of food activities. What all food we are taking depends on that. The liver will produces more cholesterol. And in some cases, our body cannot remove LDL, that means low density lipoprotein, from our blood. This is also one reason. Then certain disorders, if some people have diabetes, automatically cholesterol will come like that. Then eating high saturated fat and not having exercises, so this is also one reason. Another reason is overweight. By overweight also cholesterol will come. Then some high blood pressure, smoking, this is also results in cholesterol. Then diagnosis. By taking these medicines, we can reduce the cholesterol level, that is lovastatin, pravastatin, simvastatin, and atorvastatin. Also the niacin and some bile acid sequestrase, cholestyramine, 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 cholesterol absorption inhibitors, fibric acid inhibitors. So by using these medicaments or undergoing these type of treatments, we can reduce the cholesterol. See, fibric acid derivatives means they will lower the triglyceride levels. Also, they will lower LDL level also. And for high triglycerides and all, we are using this fibric acid derivatives. So these are the two examples for the fibric acid derivatives. In the case of cholesterol absorption inhibitors, this will lowers the LDL cholesterol. Then bile acid sequestrates. This is mainly used for to treat the high levels of LDL. We can reduce by taking this bile acid sequestrates. And then common side effects will be there, that is bloating, constipation, heartburn, etc. Also for high level of triglycerides also, we will now take these bile acid sequestrants. Then niacin. Niacin means it is very effective in high HDL, means high density lipoprotein. There will be side effects, redness of the skin, then stomach upset, headache, dizziness, our vision will be blurred, liver damage, etc. This is the lovastatin, these are statins. Usually the doctor will suggest these type of medicines. So that's for today. Thank you.